So cyanobacteria, despite being lowly single-celled organisms, have to have senses. It makes sense for them to want to know when is it light out. So they turn on all the genes that are important for collecting light and doing photosynthesis. And then when it goes dark, when the sun goes down, then these bacteria optimally, if they had brains, would want to turn all those proteins off to stop making them because there's no point having them overnight. So these scientists have identified the promoters, the DNA sequences that are in the promoters upstream of those genes that should be regulated by light and dark cycles, and they've cloned those. So they've made the firefly luciferase protein regulated now by promoters that turn on at night and turn on during the day. So what you see here is the scientists turn out the lights. So what we're looking at now is glowing, these bacteria actually glowing because scientists have put luciferase, an enzyme that cuts a chemical and produces light. And so this lawn on the left, that luciferase gene, has a promoter that contains the DNA sequence that controls expression of genes at night, hence the moon. And on the right is the same gene, luciferase, but in its promoter, where DNA elements are helping proteins bind and turn on or turn off transcription, like our Cry1 gene. Right, these bacteria produce the proteins that are important for photosynthesis during the day. So if we turn off the lights and watch the glow again, see right now proteins are binding to the genes that are turned on at night, and then now, this is time-lapse by the way, so 24-hour cycles, you get overnight genes, daytime genes, overnight genes, daytime genes. What's surprising about this? Anything? What could have occurred other than that oscillation? Okay, so lights go off. So now we're just looking at fluorescence. Nighttime genes, daytime genes. Why did the left side go dark? So once you produce this enzyme, luciferase, it starts chewing up this chemical substrate it has and releasing light. So why didn't it just stay light all the time?
It ran out of the material that was required for it to glow.
So there are a couple of possibilities. Indeed, one is luciferase is limited by the amount of substrate. What else controls this oscillation, do you think? Proteins during the day, proteins at night, proteins during the day.
Isn't it like absence or presence of lactose? In this case, would be absence or presence of light.
Well, in this case, light, unfortunately, in this case, light doesn't control directly the expression of the proteins. But there's something that does control the expression of proteins. Those are the transcription factors that are responding to light and dark cycles. So the other thing that happens besides the presence of the substrate that luciferase acts on is proteins don't last forever. We talked a couple of weeks ago about transcripts and how messenger RNA molecules, once transcription occurs and you make messenger RNA, those messenger RNA molecules in our cells don't have an infinite lifetime. At some point they get degraded, and the same is true for proteins. So what we're seeing here in this video is that those proteins, those luciferase enzymes, slowly die, essentially. They get degraded by the cell, and that's why those bacterial colonies dim and then go dark over time. And then 24 hours later, they get bright again because that is the expression, the creation of new luciferase enzyme molecules, new proteins. that convert more luciferin into light, and then they degrade as well. So that's why we see that oscillation. So that's protein degradation. Any questions about that? So this is the point for studying gene expression regulation. This is an example of gene expression regulation, as is the case for Cry1. Yeah?
How come they all do it simultaneously?
So this is a colony of millions of bacteria, and they're all coordinated.
Right.
So every bacterium is doing the exact same thing at the exact same time. Much like humans have a circadian rhythm. So it's the same question for humans. How do we mostly all go to sleep at the same time and wake up at the same time? It's genetics. This raises a really important question that I hate to have to gloss over, but if I can expand on your question, I would ask, what is it that's controlling the production of luciferase? Something is a transcription factor, a protein is binding upstream of luciferase and turning it on and making it be produced, and that's when we see the glowing. Well, then something has to be regulating when those proteins that turn on luciferase get turned on. So what's regulating the transcription factor production? Because it's the transcription factors then that turn on luciferase. So it's this chicken the egg thing. So then there has to be a protein that turns on the transcription factors that turns on luciferase at certain times of the day. Right? So that's where we're headed. Complexity.
Question, is it possible that luciferase is just like, is always, after time, it's always present even during the lifetime, but we only see it once the lights go dark?
There might be a little residual amount of luciferase, but it does turn over, that is, get degraded pretty quickly. Hence the darkness. All right. So the goal in this class and next class is basically twofold. One, to understand how mutations affect the regulation of transcription and translation, gene expression. And then, hopefully, the ultimate outcome will be that I could, say, design a mutation to have a specific effect, and that you'd be able to do that. So let's take a look at how we're doing on Socrative. So, an operon. All right. Mixed bag. Nobody watched the video. Or a third of you watched the video. Set of genes. So what's the difference between an operon and an operator? I think that's the source of this. We have two words, that's why I asked that question. We have two words that we're working with that start with oper. Important distinction. So the operon is like lac ZYA. It's a part of the bacterial chromosome that goes on because it's circular here. There's a single transcription start site, and then there are 3 genes that get transcribed together. That's an operon. It's a set of genes that get transcribed together in a single messenger RNA molecule, a single RNA molecule, prokaryotes. So that's the answer to that question. So what does this mean? If I make 3 transcripts that have lac Y, how many transcripts have lac A? They're all transcribed together. Single messenger RNA molecule gets produced. I have 3 transcripts that have lac Y, how many have lac A? 3. Yeah. So after transcription, you get a single messenger RNA molecule that has the complementary determining coding sequences for LAC-Z, LAC-Y, and LAC-A. The reason that bacteria do this is because all three of these genes are necessary for what? Which operon is this? Lac operon. What's lac? Lactose. All three of these genes are necessary for bacteria to use lactose. So, in what situation, you play bacteria, in what situation would you want to make LAC-Z without having LAC-Y and LAC-A also? There's no situation. You need all three to metabolize lactose. So you never want to make LAC-Z without having some LAC-Y and some LAC-A proteins present as well. If you want to learn more about this, I suggest that you get online or crack a textbook and look up what are the proteins that are produced by LAC-Z, LAC-Y, and LAC-A, what do they do? And you'll find out what their roles are in the cell. It's known. So this is the purpose of operons in bacteria, is that they're sets of genes that need to be all produced at the same time. Sufficient. What's an operator? Hello, operator. That's a part of the promoter where transcription factors bind to regulate whether or not the LAC operon is transcribed or not. What binds the operator? What specific protein binds the LAC operator? It's LAC repressor, and its letter is LAC-I. So I'll either call it LAC-R or LAC-I. Unfortunate. I apologize. This is the nomenclature. Don't hate. Don't shoot the messenger. The gene is LAC-I. LAC-I encodes LAC repressor. So LAC repressor binds to that DNA sequence of the operator. Questions, concerns? All right. Relationship between an effector and an allosteric site. What is an operon? Wrong question. Thank you for pointing that out. Effector and an allosteric site. Ah. What about your eyesight? Yeah, my eyesight's not doing very well either. C is correct here. An effector binds a protein, causing it to change shape. So, allosteric, change shape. What protein does lactose bind? In bacteria, what protein does lactose bind?
Isn't it LAC-80?
So lactose binds LAC repressor. So we've got lactose, we've got LAC repressor. So does LAC repressor normally bind to DNA or normally not bind to DNA? Its normal job, yes. Good answer. Very clever. What's LAC repressor's job? It's in its name. To repress. How does LAC repressor repress the LAC operon? LAC-Z, LAC-Y, LAC-A. How does LAC repressor keep the operon from being transcribed?
Doesn't it like latch onto a piece that doesn't allow polymerase to
Right. So LAC repressor, yeah, basically what you said. LAC repressor binds the operator, and that keeps RNA polymerase from getting into the promoter, binding the DNA, and starting transcription. So lac repressor is a roadblock. It sits on the DNA sequence in the promoter and prevents proteins from getting access to the promoter and starting transcription. That's in the situation where there's no lactose. What happens when lactose binds lac repressor? Up here.
It changes.
We get an allosteric change, that is other shape. Lac repressor no longer is able to bind DNA. Lactose binds, lac repressor changes shape, let's go with the DNA, and it can't bind and repress transcription anymore. What happens when all the lactose gets used up?
Then it changes back.
Right, then
back to the DNA.
this lactose molecule pops out of lac repressor, gets degraded by lac-ZYA. Actually, in truth, it's specifically lac-Z that actually chews up lactose, converts it into glucose and galactose. And now lac repressor goes back to its original repressive state because there's no more lactose around. So this is elegant. This is bacteria sensing their environment. They can tell whether or not there's lactose in their cells, and they turn on and off the transcription of the genes they need to metabolize that sugar based on its presence. So that was Socrative question 4. When does lac repressor prevent lac operon transcription? It's in the absence of lactose. You don't want the lac operon transcribed when there's no lactose around. Let's see, hopefully, oh good. Whew. So the blunt arrow is used to represent repression. The pointed arrow, or a regular arrow, represents activation. Nice work. Last one, constitutive expression. Means transcription is always on. So that's A. So all important points for continuing on with today's content. Now, so this is, which side is lac repressor on? It's a repressor. So it's up here, right? Repressor binds the operator. RNA polymerase would bind to the promoter and get recruited there, but then it can't move into the coding region, into the operon, because it's being blocked. Talked about that. So here's your turn. We know about the blunt arrows and the regular arrows. So you, by yourself or with a partner, doesn't matter, please draw the genetic pathway, the interactions between lactose, lac repressor, and the lac operon, lac-ZYA. So you should have those three terms, and you just need to draw either blunt or straight arrows between them. So go ahead and work on that for 1.5 minutes, discuss with a neighbor, and then I'm going to take your responses afterwards. Go ahead. It's going to be blunt arrows or straight arrows that connect these terms. This is simple and yet complicated. There's an important, so let's pull back together now and I'll take some responses. This is an important expression of genetic logic. So what are you most confident about in terms of arrows between terms? Lactose represses the repressor. Okay, lac repressor represses the lac operon. Are we okay with that? Its name is repressor. So how do you know that that's repressive? Right, so lac repressor stops transcription of the lac operon. So the way I think about this, in case it sounded like this was a discussion some of you were having, think about what you might consider to be the normal function or the normal job or the normal role of that protein. So when that protein is active, what is it doing? It's binding and it's stopping transcription. So its normal job is to repress. That's why it's a blunt arrow. Lactose and lac repressor. Can you explain the logic to me there? So when lactose is present,
It latches onto the lactose.
it binds lac repressor,
Changes its
changes its conformation.
and then it just flies off the end.
Right. So lac repressor is not able to do its normal job
Right.
in the presence of lactose. Its normal job is repressing. So if you repress a repressor, what does lactose do to transcription? This is not an arrow you needed to have drawn, but this is the important logic of genetic pathways. When you have a double negative, when you repress a repressor here, there's a repression of a repressor. That means that lactose activates transcription. Two negatives make a positive. Repressing a repressor makes an activator. So you could consider lactose to be activating transcription because it represses lac repressor. Yeah.
I have one question. The first part, the lactose represses lac repressor, it's okay. But I think from lac repressor to lac operon, wouldn't it be activated? Because when lactose represses lac repressor, it means it doesn't bind anymore, so it activates the lac operon.
Right, so this is the important distinction for how to determine what is the normal function of lac repressor. Yes, it binds DNA, but its role is to repress. So I totally agree, this is the confusing part. You're defining, if I understand correctly, the activity of lac repressor as being a DNA binding protein. And in that sense, lactose prevents it from doing that job. The function, though, of lac repressor is to repress. So that's the difference. And that's why this is a little bit complicated. So again, you have to be, when you discuss repression and activation, you have to be very careful about how you define whether or not you're talking about the function of a protein, what it's doing, or its effect on other proteins. Yeah?
So how, just to clarify, so the repressor doesn't it go and bind to the polymerase and prevent the polymerase from attaching, right?
The lac repressor binds to the DNA at the operator. So here's the DNA. The operator sequence, we get lac repressor binds there, and the promoter is here. And that's where RNA polymerase would bind.
But because the repressor binds, therefore DNA polymerase can't bind, right?
Yeah, it can't move, it binds, but it can't move past the operator because lac repressor's grabbed onto the DNA there and won't budge until lactose is present. It binds to lac repressor, makes lac repressor leave the operator, then polymerase can get through the operator sequence and start transcribing.
Looks like a feedback loop, right?
Ah, very much like what we heard about at the start of class. about repressors repressing repressors. So we have repressors that are degrading proteins that are involved in our circadian rhythm. That's what we heard about at the start of class. This is exactly the same idea. This feedback loop, inhibition of an inhibitor. Very salient presentation to have had today. I'm going to skip through this slide. I've just given you a summary of what lactose and lac repressor do. This is the wild type state. In the video, I talked about some different types of mutations that occur in this system to lac repressor and to the operator, right? So to the protein that binds the operator and to the DNA sequence itself. And this is my analogy for constitutive expression. If you were the unfortunate owner of one of these cars that had a recall because the accelerator would sometimes get stuck on the floor mat, bad design. This was constitutive expression, unregulatable speed. Right? This is like constitutive expression for DNA, for transcripts, and for operons. It's like the accelerator's pedal to the metal. Can't regulate transcription, it's just always happening. Transcription of the operon all the time, can't stop. It's bad for a bacterium. And there are two mutations that are in the video, the OC and the I- mutations, that are both of this type. So those are these two, where regardless of whether or not in the absence of lactose, presence of lactose, there's no difference in the state of transcription. In both cases, the OC mutations, the I- mutations, the lactoperon gets transcribed regardless of whether or not lactose is there. So somebody summarize for me quickly, 15 to 20 seconds, what happens in the OC mutation? What's the effect? What happens when that mutation occurs? I'm not talking about Orange County people. Yeah, how does it do that? It's in parentheses up there, just in case you're... Changes the sequence of the operator. Yeah, the operator is a DNA sequence that the repressor binds to. So guess what? Repressor can't bind the operator if it's not the sequence that the repressor is looking for. So a mutation in the operator prevents repressor from binding. So what happens? If you can never repress, you always transcribe. RNA polymerase binds, transcribes the operon, binds, transcribes the operon. You have unregulated transcription, constitutive transcription.
What if there's no like lactose around? Is it just efficiency that you're affecting?
Well, what happens if lactose is not around? What does lactose do to lac repressor? It changes its shape. So this is a great point. Let's draw out the situation here. We've got the operator sequence. It's got a mutation, so repressor can't bind. Repressor is going to sit up there. Okay, so then let's look at the situation when lactose is present. It's bound to lac repressor. What's repressor doing when it's bound to lactose? Bound or it's unbound? Unbound. So in the presence, it's unbound on the left side because it doesn't recognize the new operator DNA sequence. It can't bind to that sequence. So regardless of whether or not lactose is present, lac repressor doesn't bind the operator. So that's the OC for operator, constitutive. It's a mutation in the operator that causes constitutive expression. Lac repressor never binds the DNA at all. Presence or absence of lactose, either one. our last 10 minutes, I want to talk briefly about cis and trans regulation. Who's heard of cis and trans regulation before? A few willing to cope. All right. So what I want to know is, what's the effect? So you have a lack. Let's start at the bottom here. I was going to start at the top, but hell, let's change things up. Since we just talked about the lack OC mutation. What's the effect of that mutation? Changes the DNA sequence, which means transcription's always on. So you've got constitutive expression in the lack OC situation. Now this is a mutation to the bacteria. We could give the bacteria some genetic therapy. We could provide it with a new chromosome that has the wild type. That's what this column added plasmid. Remember, bacteria have circular genomes. We could give it a little tiny circular genome. We could inject this or electroporate it into bacteria, give it new DNA to try to solve this problem. So let's say we have this plasmid, and the plasmid has the LAC-O plus, the wild type version of LAC-O. What's the effect? So now you have a bacterium that has that plasmid, and then its normal genome, it looks like this. Transcription start site, there's the operon, Z, Y, and A. We have an operator upstream of that with the constitutive mutation. So we've got a bacteria now that has wild type operator, it has some mutant operator. It's got the LAC-ZYA operon, it's got LAC-I, which produces lac repressor. So you've got wild type lac repressor floating around in this bacterium. Do we still have constitutive expression? Is LAC-ZYA going to be transcribed? So assume absence of lactose. What should lac repressor be doing in the absence of lactose? Binding. So where is it going to bind in the absence of lactose? It'll bind to its wild type operator.
It wouldn't make any difference.
Which doesn't make any difference because that's
Not the DNA where the LAC-ZYA is located.
That's not where transcription happens.
exactly. So we've given it a wild type copy of its binding site, the operator, but that operator here, this DNA sequence isn't controlling anything. It's just a fragment of DNA on a plasmid we stuck into this bacterium. What's happening here, this is where we want to know what the action is. LAC-ZYA. Is the wild type lac repressor binding the mutant operator? No. So there's no effect. We tried to help the nice bacterium by giving it a wild type copy of the operator, but we haven't solved anything. That's because regulatory mutations, regulatory sequences like the operator, are these cis or trans? Cis acting or trans acting, those of you that know? Cis. Because a mutation only affects the genes that are linked to it on the same molecule together, cis. What does trans mean? Across or opposite. So which of these is going to be a trans effect? Let's say we have a bacterial genome that's got a LAC-I minus. We've got LAC-ZYA, we've got the wild type operator. What happens in the I minus mutation? Can't bind to the operator? Okay, so what happens when make lac repressor can't bind to the operator? Right, there's a mutation in lac repressor. What happens if we give it wild type on a plasmid? If we add the I+, the wild type lac repressor gene to this bacterium? Now that protein can bind to the operator and repress. So that's why this is a trans effect. The presence of that protein in the cell can go work like the normal protein would. So basically, what do we have now talking about eukaryotic genetics? What have we kind of made? You can't do this, and we don't talk about this in prokaryotes, but what's the genotype of this individual? They're like a heterozygote. Got one wild type copy, one mutant copy. In this case, providing the bacterium with the wild type copy of the protein, lac repressor, helps it work like normal. And that defines a trans effect. And what I'd like you to think about for next class, we've still got a couple minutes, but I want to wind up telling you a couple of other things, is to think about what's going to happen in this middle situation. So sketch out with the lac-I super repressor mutation. Super repressor? What happens if you give that bacterium the I+, a wild type copy of lac repressor? Will that solve the bacteria's problems with regulating transcription or not? Okay, now, a couple things. We've got a textbook reading for next class. We've got a video on Wednesday. We'll finish up with prokaryotic. We'll talk about the answer to that question I just posed. We'll move on to eukaryotic transcription regulation. We've had one Comedy Genius video uploaded so far. So if you'd like to care to go to the box folder, try to log in, see if that works, tell me if it doesn't, and leave comments. I'm hoping by the end of the semester we've got more people that submit videos to Comedy Genius so that the person that gets the most comments on their video will win some sort of a prize. And since I've got 1 minute left, I'll show you the grade distribution from the exam. So, exam information. No? Okay. So I'm going to upload the grades later today. And all I've got for you now is the grade distribution, which is better. Not that bad. Could be better, but compared to previous exams, people are moving up. So that's good. Again, hard to say grade distributions, but I'm going to say that that's maybe that might be the situation. I'm kind of fuzzy about a grade of 3. So I'm thinking if you see when you get your grades this afternoon, if you've got a 3, 2, or 1, that's not so good. If you're above 4 and above, that's pretty good. Well, so you realize I write hard exams, and I've given you, especially the first question, which you probably don't remember. That's okay, we'll talk about it soon. You've never seen that question before. We never did anything like that. So nobody got, one person got that question right, I think.